Today, we are going to introduce a third-generation sequencing technology. In this video, we will provide a brief overview of the working principles of third-generation sequencing and fundamentally dissect its advantages compared to the previous two generations of sequencing technologies. Furthermore, we will discuss its various applications in clinical diagnostics, its unique features and advantages. Finally, we will provide a detailed introduction to the highly acclaimed single-molecule real-time sequencing within the realm of third-generation sequencing. Now, let's commence with this informative session. Third-generation sequencing is an emerging DNA sequencing technology with prominent platforms such as Pacific Biosciences and Oxford Nanopore. Unlike traditional first-generation Sanger sequencing and second-generation NGS, next-generation sequencing technologies, first and foremost, third-generation sequencing technology can generate longer DNA fragments, making it better suited to address complex genomes and repetitive sequences, as well as enabling whole chromosome sequencing. Secondly, third-generation sequencing is typically single-molecule sequencing, which means it can directly determine the sequence of individual DNA molecules without the need for PCR amplification. This helps reduce sequencing errors and eliminate potential biases. Thirdly, unlike traditional batch processing sequencing, third-generation sequencing technology can sequence DNA molecules in real time as they are being read. This means that data can be generated in real time without the need to wait for the sequencing run to be completed. Third-generation sequencing technology offers numerous advantages in clinical diagnostics. For instance, in rare disease diagnosis, the long-read capability of third-generation sequencing is particularly well suited for detecting gene variations such as structural changes and repetitive sequences. In cancer genome research, researchers can use third-generation sequencing to comprehensively analyze complex variations in tumor tissue, aiding in the discovery of new therapeutic targets. So how does third-generation sequencing actually work? Currently, third-generation sequencing technology can be broadly categorized into two types: single-molecule real-time Smrt sequencing and nanopore sequencing. Let's delve into the details of the single-molecule real-time sequencing platform. Single-molecule real-time sequencing, developed by Pacific Biosciences, involves the labeling of each DNA base with a fluorescent dye during the DNA synthesis process. After the DNA template is captured by the DNA polymerase, the four different fluorescently labeled bases randomly enter the detection area through Brownian motion and associate with the polymerase. The time it takes for the base that matches the template to form a chemical bond is significantly longer than the time other bases spend in the detection area. Therefore, by analyzing the duration of fluorescent signals, one can distinguish between matched bases and free bases. By plotting the relationship between the four fluorescent signals and time, the DNA template sequence can be determined. One of the core technologies of Smrt sequencing is zero-mode waveguide, ZMW. A zero-mode waveguide is a hole with a diameter of only 10 to 50 nanometers, which is much smaller than the wavelength of the detecting laser. Therefore, when the laser is directed at the bottom of the ZMW, the laser cannot pass through it. Instead, it undergoes diffraction at the bottom of the ZMW, illuminating only a very small area. DNA polymerase is then immobilized within this area. Only within this region can the fluorescent labels carried by the bases be activated and detected, significantly reducing background fluorescence interference. Each ZMW immobilizes only one DNA polymerase, and when a ZMW binds to fewer or more than one DNA template, the sequencing results generated by that ZMW are filtered out in subsequent data analysis, ensuring that each available ZMW represents an individual DNA synthesis system. A total of 150,000 ZMWs are grouped together on a chip, forming a single SMRT cell. Within a single run, the PacBio RS2 sequencer can simultaneously sequence 8 SMRT cells, producing 3.2 gigabases of data. Another core technology of SMRT sequencing is the fluorescent label attached to the 3' phosphate of the nucleotides. During DNA synthesis, the 3' phosphate bond is cleaved as the DNA strand extends, discarding the label and reducing steric hindrance in DNA synthesis. This allows for continuous DNA strand synthesis, leading to longer sequencing read lengths. In contrast, in second-generation sequencing technologies, fluorescent labels are attached to the 5' methyl group of the nucleotides. During the synthesis process, the fluorescent labels remain on the DNA strand. As the DNA strand extends, it creates three-dimensional spatial hindrance, leading to misreading when the DNA strand reaches a certain length. This is one of the reasons that limits the read length in second-generation sequencing. SMRT sequencing maximizes the activity of DNA polymerase and represents a polymerase reaction system that closely mimics the natural state. In the real-time monitoring system, DNA strands are synthesized at a rate of 10 bases per second. The entire process, from library preparation to sequencing, is completed within 2 days. It's worth mentioning that PacBio utilizes circular DNA molecules and sequences them in a looped fashion. Consequently, DNA polymerase can synthesize DNA strands by looping multiple times on the same template. So, we conclude our presentation for today. Have you gained a deeper understanding of third-generation sequencing and single-molecule real-time sequencing?